but I'll take this as step one. All right. Can you see my talk?
Yes, sir.
Perfect.
All right. Fantastic. So as Christina said, I'm going to be speaking about contact lens drug delivery. These are my financial disclosures. I'm an inventor on the contact lens technology, and also I am a founder and a consultant for a company that was founded to commercialize the technology. And this is the disclosure that was developed by partners, so I could share it to everyone. All right, so let's take a step back. Most of the medications we give are in the form of drops. Only about 1 to 5% of the medication in a drop goes into the eye, though, so not very efficient. That said, of the medication that gets into the eye, 90% of it goes through the cornea. Okay? So the cornea is really important. It's a conduit for drug delivery into the eye. What's ironic here is that the cornea only accounts for 5% of the ocular surface. So even though the cornea is 5% of the ocular surface, 90% of the drugs are going through it from any drop that's placed in the eye. So, there's many forms of drug delivery that has been proposed, but if the cornea is so important for drug delivery, why not target the cornea? Well, that's what we've done. So in a sense, the cornea has a home field advantage when it comes to drug delivery. And a contact lens, since it sits on the cornea, would be more or less a way to facilitate that. There's some advantages with the contact lens, but the concept isn't new and there's many challenges. One of the challenges is getting enough drug into the lens. So you're dealing with a finite amount of space. You don't have a lot of area to work with. Think about Manhattan. So you don't have a lot of space, so they build up. Here we can't really do that. We're confined to the dimensions of a contact lens. Next, you need to have a mechanism for controlling the release. Most contact lenses, if you were to soak them in a solution and place them on the eye, the drug will come out very quickly, half hours. So what we've done is we've enabled a system that allows us to load a lot of drug and yet release in a controlled manner. What we have is a very thin drug polymer film situated in the periphery of the contact lens. The contact lenses are similar dimensions to commercially available bandage contact lenses. They can be made with or without vision correction, and they're made completely with FDA-approved materials. So I'm going to focus on ocular inflammation. This is a dexamethasone-eluting contact lens. And this is the drug flux in rabbits compared to drops. Not just any drops, but drops that were placed every hour for 8 hours. And these were the drug levels we got on the cornea. In comparison, we placed a contact lens on the eye. And at various time points over a week, we euthanized the animals and looked at the drug levels. Now, mind you, this is a log scale. And so you can see that we're getting much more drug into the eye. And not only can we get the drug into the eye, but it's staying there for a long period of time. And this is just with one contact lens. We get to the cornea, and even at the retina, we're getting significant levels into the eye there. So what about efficacy? We know the drug's getting there, but does it work? We've done 4 different models of efficacy studies. One is on corneal neovascularization. Another was on corneal haze. Another one is on uveitis, and also retinal vascular leakage. For today's talk, since we are mostly focused on the front of the eye, I'm going to focus on the corneal vessel experiments. So for this study, what we did is we placed sutures along the cornea on the inferior and superior aspect of the cornea in a figure 8 fashion. We took images at the time, and then we initiated treatment. We repeated the imaging on day 5, and then on day 7, the animals were euthanized. We then removed the cornea with a button and divided it in half. Half of it we used for PCR, half of it we used, and that was to look at the VEGF levels, and the other half we used to look at the CD45 cell frequency, so inflammatory marker. And so here's some images here. With a dexamethasone-eluting lens, you can see that there's almost no neovascularization present. Same with drops. Drops is very effective at preventing the neovascularization. However, with no treatment or with the vehicle lens, you have these vessels that have gone over and started creeping from the limbus. In fact, you can't see the upper half of the figure 8 of the suture on either of those. So from a picture alone, this was very effective at preventing the neovascularization. And we measured the area. This was at day 5 in white, day 7 in black. And we can see that we had significantly less, there's a p-value up here, but significantly less neovascularization compared to no treatment and the vehicle. What about inflammatory markers? Here, the CD45 cell frequency, we had very similar to the drops and significantly less than the untreated or vehicle. And the levels that we measured were very similar to the contralateral eye, which did not have any sutures. What about the VEGF levels? We looked at it 2 different ways, the VEGF KDR and the VEGF-A, and we found that we had significantly less VEGF KDR than the vehicle or the untreated. We weren't significantly less than the DEX drops, but nevertheless we were it was a fairly small study, so I'm not sure if we can differentiate effect with such a small study. But the VEGF-A, actually the drops had lower levels than our lens. This is biology. You never know what you're going to get. And one of the surprising things we found was that we looked at the corneal thickness. I figured that folks like Debbie Jacobs would ask me about the cornea thickness and how this affects things. And so we looked at that. And here we measured it with OCT, and we can see the no treatment on day 0, day 7, the dexamethasone drops, our dexamethasone lens, in the vehicle. And just from this figure alone, I'm going to go back here for a second. From figure alone, you can see that the vehicle was significantly greater, result in a greater thickness in the cornea. And when we look at the measurements, we see the same thing. And this was the change in central cornea thickness at day 7 compared to baseline. And what's interesting is the dexamethasone lens had significantly less thickness than the vehicle. Not only that, significantly less than even no treatment. And the drops helped a little bit too, but not as much as the dexamethasone lens. We did a 4-week biocompatibility study. We did this in order to study the effects of the lens so that we can use it in humans. And what we found is that once again, the effects didn't wear off necessarily. This was a lens repeated every week for 4 weeks. And our box plot here overlaps 0. So there wasn't any significant change in the cornea thickness compared to a commercial contact lens, which had significantly increased corneal thickness. So it seemed as though, no one knows exactly why, but it seemed as though the dexamethasone in the lens prevented corneal edema. So I'd like to summarize where we are so far. I've only showed you one of the efficacy studies. Our other studies have been published, and I can certainly send you the link if you'd like. What we've shown is for dexamethasone-eluting lens, we have controlled release. We have drug flux that was superior to hourly drops, a lot more drug out in the cornea than the hourly drops. We've shown efficacy in 4 different animal models, and we have corneal edema that resulted from the control, the vehicle that is, but it really didn't show that we had increased corneal thickness. It was unchanged. The human studies are actually this year. We have consented our first patient, and we should be having some data in the next month. And our indication we're looking at, you might find this hard to believe, but working with my retina colleagues, and we're looking at cystoid macular edema. And so we should have some data this spring on the effect of that. So other things we're looking at that I'm really excited about. I didn't go into it here because we haven't published our data yet, but we have a moxifloxacin-eluting lens that we were able to show was effective at treating MRSA keratitis that was resistant to moxifloxacin. So here we're able to use a drug more effectively instead of having to rely on other drugs of last resort like vancomycin. So let me pause there, and you can see there are all the different contributors I'd like to thank. And this would be a good time to answer some questions if there are any.